For chapter one of microbiology for this class, it's pretty straightforward. We're just going to go over some of the key principles of microbiology. We're going to talk a little bit about a microscopy, and then we're going to talk a little bit about our defense system, right? So you have your innate as well as your adaptive immune defense system. So we're going to go into details on all those different elements. Throughout the semester, we're going to go into much more detail over, especially description of different types of microorganisms. Not so much microbiology. Microscopy. We're going to talk a little bit about microscopy, only insofar as it pertains to what microbiology is. And microbiology effectively is anything that you cannot truly see with a naked eye. There's a couple exceptions to that rule. That's when we start getting into helminths. So those are like parasitic worms. Generally speaking, a parasitic worm is something you can indeed see with your naked eye. So think about like a Tinea solium tapeworm or like a liver fluke. Those kinds of things you can actually see with your naked eye. But the reason why it falls under the umbrella of microbiology is because generally speaking, when you get infected with one of those organisms, you're ingesting the egg. So you can't see the egg when you ingest it, right? You can only see the egg underneath microscopy. But once it gets introduced into your system, and then the ovum matures into its full-sized helminth, then you can actually see those with the naked eye. And I'm going to show you a couple of videos of those tonight, some of which can be rather distressing. So in other words, microbiology is effectively the study of everything that you cannot see with your naked eye. Microscopes are indeed going to be required, and there's several different types of microscopy. The most ubiquitous and the most commonly used microscopy in microbiology is going to be light microscopy. We are going to do light microscopy on week 3 of our labs. Week 3 is going to be our gram staining lab. We're also going to do aseptic technique. So that's where we transfer bacteria from one sample and inoculate a sterile medium using inoculation loops, open flame, things like that. So we're going to learn how to do aseptic technique. And then we're going to do gram staining, which is kind of like the gold standard way for us to actually visualize bacteria. We do gram staining for gram positive and gram negative organisms. And then that's, you'll see the different colors. We're going to go into those details a little bit throughout the semester, but gram staining is probably the most commonly used staining method for bacteria. When you start getting into viruses, then you start getting into things like electron microscopes. So there's scanning electron microscopes and there's transmission electron microscopes. Both of those are going to be referred to as SEM or TEM. And those are going to be for things that are way smaller than bacteria. Bacteria, of course, are going to fall under like single cellular type organisms. Viruses are going to be much smaller than those single celled type either organisms or single celled structures that you're going to see in eukaryotic cells. So here's an image that I included. The image on the top right is going to be your neuromuscular junction. So those are going to be the nerves that innervate your muscles. And in order to visualize that, one of the best ways to do it would be through a scanning electron microscope. So this is a scanning microscope. You can actually see the surfaces of different structures. I'll show you a couple of other images of what that looked like. Transmission electron microscopes, you see right through the structure. It's really good for visualizing things like flagella, for viruses, for different organelles in a eukaryotic cell, for example. But for scanning electron microscopes, you can really see the surfaces of different structures, and you can see those surfaces at a very small scale. So we already kind of talked about this, how cells, of course, are going to be, you can't really see it with a naked eye. So any of those structures you're going to have to see with microscopy. If you use light microscopy, you're able to visualize things up to 1,000 times their size. Now, when we work in the lab on week 3, the microscopes that we use in lab, they have 2 different lenses. They have the optic lens and then they have the objective lens. So those are two different lenses that combine, that work in concert. The optic lens is going to be at 10x, so it's 10 times the size. So if you looked at anything with the optic lens, everything would look 10 times the actual size. The objective lens, which actually rotates, there's multiple different lenses on those. Depending on depending on how good your microscope is, you can go up to 100x. Some don't quite go as high, but the ones that we use, we do have ones that do go up to 100x. So you go up to 100x, you're multiplying 100 times the optic lens, or sorry, the ocular lens. And so it's going to be 10 times 100. You're looking at something that's going to be actually about 1,000 times the actual size. Now, speaking of microscopy, you guys are going to have to know a little bit of your historical figures throughout this class. The first of which is going to be kind of like he's like the godfather of microbiology. His name is Anton von Leeuwenhoek, and he was a microscope builder. And what he was doing back in the 1600s was he was actually taking tooth scrapings off of his teeth, and he was looking at pond water underneath the microscope. And I included this image in the background here of his little handheld microscope. Don't ask me how it works. We don't really use these anymore nowadays, but it's a very rudimentary microscope. But what he was able to do, von Leeuwenhoek, was he was able to actually visualize the first microorganisms. He referred to them as animalcules. He also referred to them as wee beasties. So he didn't really publish his material. His findings were sort of like discovered after the fact. He did talk about his findings of like those animalcules in his journal notes. So not really something that he published, but he was the first one to actually visualize those microorganisms. So very interesting dude. We'll get back to him later, but there's several different key figures in microbiology, but Anton von Leeuwenhoek arguably is one of the first and foremost microbiologists because of the fact that he was the first one to discover those tiny microorganisms. Here's an image on the top right. That's another scanning electron microscope image. Do any of you guys know what this organism is? We're not going to study this in microbiology, but it is it's microbe basically. It's this very, very tiny organism.
It's a water bear.
It's a water bear, yeah. So a tardigrade or a water bear. Tardigrades are really cool. The reason why they're super cool is because they are considered extremophiles. Now, when we use that term extremophile, we're usually referring to archaea, and we're going to talk about that a little bit later. Prokaryotes are going to be subdivided into two different categories. One is going to be bacteria or Bacteriaceae. The other one's going to be archaea. And archaea are going to be those types of organisms that can live in very extreme environments. I'm talking like hydrothermal vents at the bottom of the ocean, those quote unquote black smokers, or in places like Yellowstone National Park, where you see the geysers and they have all the crazy colors. Those are all archaea, right? And they can withstand extreme environments. They can withstand extreme temperatures, high pressure. Now, albeit tardigrades / water bears are not prokaryotes, they're going to be falling under that extremophile category. The reason being is because tardigrades are able to withstand very extreme temperatures. They can also extreme harsh conditions in terms of pressure as well as nutrient deprivation. Now, a very interesting thing that NASA did. NASA actually took a bunch of tardigrades. They put them on a rocket ship and they sent them off into outer space. And they brought those tardigrades back, and they were actually still able to survive. So they lived in not only extreme temperature of cold, right? So extreme cold temperatures, but also the vacuum of space. So they're a very, very interesting organism. So that's something you might want to look at with a scanning electron microscope. You could look at that with light microscopy as well, but so that's an example of SEM. With scanning electron microscope, you can see things up to 100,000 times the size versus a light microscope. You're looking at something about 1,000 times the size. And then at the bottom right here, those are images of actual atoms. And those images are produced by looking through an atomic probe microscope. And so you can see much more minute more minute structures using an atomic probe microscope. So let's go ahead and talk a little bit about the key elements of microscopy. This is particularly going to be important for us when we get into week 3, when we start doing light microscopy and gram staining. So light microscopes are going to be involving a couple different elements. So the two major ones are going to be both magnification and resolution. Now, with magnification, obviously, that's just going to be the, effectively the size of whatever it is that you're looking at. And then resolution is going to be involving clarity of whatever object you're looking at. So we also refer to resolution as resolving power. And resolving power is going to be involving a couple different things. We'll talk about that in a second here. But resolution effectively is the way for you to be able to identify or distinguish between two separate objects. The lower the resolution, the more blurry an object is, right? So you're not able to distinguish between two separate objects very well. Now, if you have a greater resolution, then you have less blurriness, right? And so you're able to actually identify between two separate objects. So we're going to go into that a little bit later. But this image, this is from your textbook. It breaks down effectively everything we can see with our naked eye, all the way down to things that we can only see through like an atomic probe microscope, right? So with naked eye, you can see all the way down to frog eggs, human eggs. Once you start getting into cells, cellular structures, whether it's a eukaryotic cell or a prokaryotic cell, then light microscopy is going to be ideal for that. If you start getting smaller than cells, if you're looking at subatomic, if you're looking at like organelles, for instance, or if you're looking at proteins or viruses, things like that, that's when you start getting into electron microscopy. So let me move on here. So let's talk a little bit about light microscopy. So we talked about how with light microscopy, we're going to be doing things like looking at prokaryotic cells, so single cellular organisms. So on the bottom left here, I included a couple images of what some of those single cellular organisms could look like. So for instance, gram-negative rods like E. coli, for instance, or Proteus. Sometimes they have flagella. You can actually see the flagella on light microscopy. But as I included here on the top right, you can also look at histological tissue samples too. Now, if any of you go into fields like oncology, if you take a tissue biopsy, a little sample of, something that looks like, might be potentially carcinogenic. You send that off to the lab. They observe under light microscopy if there's any sort of abnormal cells in that tissue sample. And so you can look at things like cancerous cells on light microscopy. As I mentioned earlier, light microscopy allows you to see things up to 1,000 times the actual size. One of the important elements of light microscopy is going to be staining. And then on week 3, we're going to do gram staining. There's a couple other different types of stains too, but gram stainings is kind of like the gold standard. With staining, you're actually able to differentiate between the cells that you're looking at and the background, right? Because what are the color of cells? If you looked at a cell underneath a light microscope, what do you guys think it would look like? Do you think it would look clear? It would look clear because cells are going to be mostly made up of water, right? And because of that, you're not able to really see the difference between the actual cell itself and the background. And so when we do histological slides, when we look at bacteria underneath the slide, we're going to have to stain those cells so that you're able to actually develop contrast to differentiate between the cell and the background as well. So these are all different types of microscopy. On the top left, that's what an unstained specimen would look like. You can't really see it too well. I mean, you can sort of tell that there's something there, but you're not able to differentiate between different types of structures. So you can't really see the nucleus, you can't really see the cytoplasm and like the contents within the cytoplasm. But then if you start staining and then doing different types of phase contrast, then you can actually visualize those cells really well. There's also fluorescence stains, right? So you can put fluorescent compounds onto a sample, and then you can actually be able to see, visualize different regions in the cell lighting up. And then transmission versus scanning electron microscopy. That's going to be where you can really see a greater amount of detail in different structures. So these are the different types of electron microscopy, the major types. The one over here on the left, that's a bunch of different types of images of pollen. Those are all visualized under scanning electron microscopes. So you can really see the surface structures very well with the scanning electron microscope. With transmission electron microscopy, you're mostly able to just see right through whatever it is that you're looking at. So this is an image of a flagellum, and flagella are going to have it's like 9 +2 ratio. So you have like these 9 different regions surrounding the structure. Let me turn on my marker over here. So here's 1, 2, 3, 4, 5, 6, 7, 8, 9, and then +2. You have those 2 right there in the middle. And flagella are going to be really important, especially for bacteria, to be able to mobilize, right? So it's going to provide the ability for motility. You'll see that in bacteria. You'll also see flagellation in certain eukaryotic cells as well, right? So like Giardia is going to be something that we're going to talk about later throughout the semester. Giardia is going to be a flagellated organism. So those are different things you can see with electron microscopy. Here's another slide on that. So here's an E. coli cell with a bunch of flagella coming off of it. So here's the E. coli itself. And then you have a bunch of flagella coming off, and the flagella provides motility. And then here's just a schematic of that 9 +2 image of what the actual flagella looks like. So fun stuff. And then here's another image from your textbook that goes all the way through things that you can visualize through your naked eye, all the way down to things that you require, like, subatomic type imagery. So cool. So let's talk about organisms. Now, in this class in microbiology, we're going to talk about not only organisms that cause disease, but we're also going to talk about organisms that don't necessarily cause disease either. So for the most part, actually, here's a question I'll pose to you guys. Do most microorganisms cause disease? Do most bacteria cause disease? Do most?
No.
Okay, awesome. Exactly. So most microorganisms do not cause disease. If they did, we would be in a lot of trouble, right? Because we don't do this lab in this class, but there's a lab that I do with my other campus. It's called the Ubiquity Lab. And ubiquity just means that's something you can find everywhere in the environment, right? So bacteria, viruses, those kinds of organisms are going to be found throughout the environment. Fungi, especially, like fungal spores, can exist 7 miles above the atmosphere. So you can see them all over the environment. Most of them do not cause disease. However, some that don't cause disease to begin with could potentially cause disease under certain circumstances, right? So those are what we call opportunistic infections. I'm sure you guys probably heard of that before. So take, for instance, Staph aureus, or even like Streptococcus, Pyogenes, or any of the strep species that you find in your mouth. Most of the time, those organisms do not cause disease. If they start to get out of hand a little bit, maybe potentially you might get like dysbiosis, right? Then you could potentially see organisms that live on our bodies beginning to become opportunistic. A really great example of that would be Clostridium difficile, which we're going to talk about quite a bit. Have you guys ever heard of C. diff diarrhea? Okay, most of you guys that are working in the hospital right now have certainly heard of C. diff diarrhea. C. diff is something that you're going to find naturally occurring in your GI tract. Now, generally speaking, you have a lot of bacteria in your GI. Most of that big bacteria helps things, to do different metabolic activities like break down nutrients that you consume, or they also help to tame bad bacteria. They help to keep bad bacteria at bay, right? Now, if you get dysbiosis, which is most commonly caused by taking broad spectrum antibiotics, which we're going to talk about later on the semester. So, say for instance, you get strep throat, you take a broad spectrum antibiotic, all of a sudden you kill off your good bacteria. So now you kill your good microbiota, and then now you have certain organisms that can run rampant. Clostridium difficile is a really good example of that. The reason why it's called Clostridium difficile is because it's difficult to treat. C. diff diarrhea is a really nasty organism that when it causes disease, it arises from antibiotics. How do you treat a bacterial infection that arose from antibiotics, right? So it's kind of like a double-edged sword. You're dealing with something that arose from antibiotic treatment. That being said, do you guys know what the gold standard treatment of C. diff diarrhea is? Does anybody know? And has anybody here worked in GI?
Yeah,
Fecal
isn't it like a
transplant.
fecal?
Transplant. Awesome. So a lot of you guys already knew that fecal transplant is the gold standard. When I was working in internal medicine, I was talking to a GI doc, and he explained to me exactly how the fecal transplant is done. So what happens is you get either a relative, a family member, or you get somebody that you're really, you're close to, like a good friend. You have them bring a blender to clinic, and then you have them crap in the blender, a blender that they obviously don't care about throwing away. And then after they do that, they blend everything up and they encapsulate everything, and then you do a suppository of the other person's fecal contents. And because of that, you're introducing the good bacteria from the other person, the donor, back to the host. And now you've introduced good bacteria into the host. And so that is how a fecal transplant is conducted. So
I can't tell you I ever liked that whole process or even liked hearing it. It's just horrible.
Yeah, I quite frankly, I don't really understand why anybody would ever want to go into GI. It's I don't think it's for the faint of heart, or it takes a special kind of person that's into it. So that's the fecal transplant. By the way, there's actually some really cool studies out there that talk about how recipients of a fecal transplant, their metabolic activity will actually change. It will actually change in accordance to the donor. So a lot of our metabolic activity is actually dictated by our microbiota. So that's a fun fact. So anyways, we talked about good bacteria. So that's your normal microbiota, your normal flora, right? And then we talked a little bit about how that normal flora can become pathogenic, right? And so when we talk about pathogenicity, we're mostly talking about the germ theory of disease. We're going to get into some of the history with like Louis Pasteur and guys like that. The folks that were really big in disputing spontaneous generation. That was kind of like a big theory back in the 19th century. Everything like predating the 19th century. The 19th century is when the germ theory of disease kind of became the gold standard, right? Before that, they thought spontaneous generation. In other words, you would leave like a steak or something or food on the countertop. All of a sudden, maggots start producing out of the food. People thought that those maggots came out of nowhere, right? But what wasn't well understood was that a fly had to land there, lay the eggs, then all of a sudden those eggs hatch, turn into worms. So we're gonna talk about spontaneous generation versus the germ theory of disease later on. But that being said, gold standard right now is the germ theory of disease. So we just basically talked about how a lot of our good microflora, right, microbiota, those are going to compete for nutrients, and they're going to be able to actually keep all those pathogenic organisms at bay, right? So competition is going to be one of the ways that our own organisms are able to fight off pathogens. And then on top of that, those microbiota that we have, especially in our GI tract, are going to help with our immune system response, as well as aiding in digestion, right? Because a lot of the bacteria you find in your GI tract are able to break down things like complex micro or macromolecules so that you can actually digest those and absorb them in your GI tract. So, in this class, we're going to be focusing on two broad categories of organisms. One are going to be prokaryotes, and then the others are going to be eukaryotes. So prokaryote means before a true nucleus. Karyote refers to the nucleus. But I want you also to think of prokaryotes as things that do not have a membrane-bound organelle. So let's think about a couple of examples. Mitochondria, Golgi apparatus, endoplasmic reticulum, vacuoles. Those are all going to be membrane-bound organelles. So you're not going to find those in prokaryotes. You're only going to find membrane-bound organelles in eukaryotes. And when I say eukaryotes, I'm thinking like you and I, right? We're eukaryotes. Also, plants are eukaryotes. Fungi are eukaryotes. And protozoan organisms are also eukaryotes as well. But in terms of prokaryotes, they don't have a membrane-bound nucleus, but they also don't have membrane-bound organelles. The two major categories are going to be bacteria and archaea. Back in the day, and I don't know what exact year it was, but archaea used to be referred to as archibacteria. We don't refer to them as that anymore. They sort of were kind of lumped up together with bacteria, but now they're just like a totally separate class, right? But they are very similar to bacteria in many different regards. So we're going to talk about those different details in a second here. So here's a good phylogenetic tree that demonstrates, the ways that these different organisms are kind of separated out. So you have bacteria and archaea. We're not really going to go into archaea so much in this class. The reason being is that as of right now, we don't really know of any archaea that cause disease. And because biologists and the medical community and humans in general, we're kind of selfish, we like to think about things that affect us negatively. Since archaea don't really affect us, we don't really study them as much. So for the most part, we're going to be talking about bacteria when we're talking about prokaryotic organisms. And there's several different classes. We have your gram-positives, you have spirochetes like Treponema pallidum or even Borrelia burgforderi, syphilis, as well as Lyme disease, respectively. Cyanobacteria. Most of the ones that we're going to talk about are going to fall under gram-positives versus gram-negatives. And then when we start talking about eukaryotic organisms for this class, we're going to focus on fungi towards the end, so things like Candida albicans, for instance. Those are going to be some of the ones we talk about. We're also going to talk about diplomonads. So Giardia lamblia is a diplomonad. It's a flagellated type of protozoan organism. We're going to talk a little bit about Trichomona. So these are all different organisms that we're going to talk about throughout this class. And here's another good breakdown of the phylogenetic tree. So fungi, Animalia, Plantae, those are going to be your major eukaryotic subcategories. Cool. Now, before we continue on, do you guys have any questions? No? Okay, good.
Dr. Clarkson, do you mind if I jump in?
Yeah, go for it.
Sorry, it looked like you were in a transition there, so I was going to take the opportunity.
Oh, hey, how you doing, Gina? Good to see you.
Hi.
Class, this is Gina. She assistant dean, correct?
Correct.
Yeah, so Gina is going to be working with Dr. Tara. Dr. Tara, she's going to be the dean. Gina is our assistant dean. So go for it.
So wonderful to meet all of you. I see some familiar faces. Hi, Kyle. Hi, Bonnie. Yes, I am the assistant dean. I also teach English, so you might have me for an English class, or maybe you already have. But my office is on the third floor here at the Tempe campus. If you ever want to drop in and talk, or you have any questions, you'll find me there or around. So I just wanted to take a moment. This is really an exciting class. You're not even an hour in, and we're already talking about fecal transplants. There has to be another way. Dr. Clarkson, there has to be another way.
It's one of my favorite things to talk about because it's so absolutely atrocious.
Just before dinner, Yeah,
that's there you go.
All right, but this is a fun class. I'm enjoying it already. but I just wanted to remind you, when you're on site with Dr. Clarkson, or even when you're, in a Zoom session with him, make sure you give him your full attention. Cameras on if you're in Zoom. You want to try to be in a distraction-free environment, if at all possible, so you can give your full attention. But it looks like so far so good. Everybody's looking great. I can see all your beautiful faces. And if for any reason, there's something going on and you can't be on camera, just please communicate that with Dr. Clarkson so he knows what's going on. Okay. I also wanted to let everyone know that if you haven't done so already, if you haven't gotten your level 1 fingerprint clearance card, we're having a fingerprinting event here at the Tempe campus on Monday. So that's Monday, January 9th from 3:30 to 6 p.m. And Dr. Clarkson, I posted an announcement. I hope you don't mind, but I posted an announcement in your classroom for all your students to see.
Awesome.
And it has information about the event, what you need to bring, how much it all costs. And then CJ Burnt, our student success coordinator, is really kind of our go-to person with all things fingerprint clearance card related and all things Castle Branch related, which is the system where you'll upload your fingerprint clearance card, and it houses all of that information for us. So you'll want to make sure you have your Castle Branch account going. But in any case, Monday is a great day to come to campus, again, between 3:30 to 6, and take care of that if you haven't already started the process. Okay. Now, what are your questions?
No questions. Wow.
No questions. Okay. Amazing. All right. Well, if you do have any questions, you can reach me. But again, for fingerprint clearance card questions, CJ Burnt is a wonderful resource for you, and she'll help you with uploading things to Castle Branch, with just understanding the whole process. It's not too complicated, but there are a few steps involved, and it can be a waiting process. Once you send off your fingerprint, it takes a little while to get that card back. So you want to make sure you're giving yourself enough time, because you do have to have that completed before you start core, long before you start core. I think ideally within the first semester of being a student. But in any case, I just wanted to let everyone know, again, that's next Monday, January 9th, 3:30 to 6. And yeah, I'll leave you back to all things biology related.
Awesome. Are you sticking around?
No, I'm going to get going, but I hope you all have a wonderful evening and Happy New Year.
Happy New Year to you too, Gina. It's good to see you.
Thank you. It's good to see you, Dr. Clarkson. I'll talk to you soon.
All right. Have a great night. Take care.
Bye-bye.
Rock and roll. So now this slide is really important. Make sure to make note of this, because I will take points off on your guys' written essays if you guys do not do proper binomial nomenclature. Now, a good way to remember all the different, subcategories is this mnemonic: King Philip came over for good soup. The only thing I really care about is the good soup, right? Genus and species. That's the only thing I really care about in this class. Now, when you do binomial nomenclature, genus is always going to be capital Ox, right? And when you first write your genus, write it out in full name. A good example, Escherichia coli, right? When you're writing about a common foodborne illness is Escherichia coli. Then later on, you can write, yada, 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 E. coli, right? So it'd be E. coli. So, but every time you first write the genus, you write it out and it's full, right? And then from then on, you can just abbreviate genus with the first letter, but you're always going to have genus capital Ox, and then species is always going to be lowercase, and everything is going to be italicized. Now, you guys aren't going to be writing anything by hand in terms of your assignments, so you're not going to be turning in anything to me written by hand. But if you were to write a genus and species by hand with binomial nomenclature, you underline everything, that's genus and species, right? Otherwise, if you're typing it out, it's going to be italicized. So please, please, please make sure to italicize your genus and species whenever you guys write about bacteria. So that's going to be a very important thing. Otherwise, you will lose points on that. Not a lot of points, but so eukaryotes.
Sorry, I have a question.
What's up?
So, is that with everything in the classroom? Like,
I'm sorry, I couldn't hear you. What?
look, and is that everything? Like discussions and everything whenever we're putting
Everything. Anytime that you write a genus and species name of bacteria, of protozoans, right? Fungi, right? You're always going to do genus, cap locks, and then species, lowercase, everything italicized. So Giardia lamblia, that's a good example of a prokaryote. A protozoan. Giardia lamblia.
like in discussion?
Anytime you write.
Okay.
Anytime you write something, discussions, posts, anywhere that you write genus and species, you always have to italicize it.
Or we'll lose points.
Or you lose points. I mean, in general, like in the scientific field, like that's just kind of the standard. It's expected that you do italicized genus and species. That's basically kind of expected. So Giardia lamblia, Candida albicans, Coccidioides, immitis, anything that's going to be genus and species, right? So it's going to be italicized. Cool. So eukaryotes. So the reason why I have Uncle Sam here is because that's you, right? So you are a eukaryote, along with fungi, plants, protozoan as well. But cell membrane, you're going to see cell membranes also in prokaryotic organisms too, but we're going to talk about the differences, right? There's cell membranes, but there's also going to be things like peptidoglycan cell walls in certain prokaryotes. So you're going to see a cell membrane that's selectively permeable. It's going to allow for protection of the inner components of the cell, right? It's going to allow for the storage and the transfer of things like nutrients and waste. And then the nucleus, that's where that word karyote comes from. Karyote comes from the word nucleus. And that's going to be where you find your DNA, right? So you're going to find DNA inside the nucleus. In the prokaryotes, it's not going to be a nucleus, but it's going to be a nucleoid. You're going to see a bacteria. I'm just going to draw it right here. So you have like a little bacteria cell. You have all the little, DNA hanging out. It's not going to be contained within a nucleus, but it's going to be it's going to be called a nucleoid. That's going to be the term for all the genetic material. So that's the difference between prokaryotes versus eukaryotes. So eukaryotes, true nucleus, you see membrane-bound organelles, right? They're going to be a lot more complex. And then you're going to see all different types of organisms. So fungi, algae, protozoa, and helminths. Those are all going to fall under eukaryotic organism. For the purposes of this class, we're going to talk about fungi later on. We are not going to talk about algae. We are going to talk about protozoans. Some of the most significant infections in human history are protozoan infections. Can you guys think of one off the top of your head? What's like the most devastating infection that has basically destroyed a really large population of humans throughout history? Do you guys have any idea?
AIDS or HIV? Is it AIDS or HIV?
Throughout the 20th century, HIV/AIDS is considered like one of the most significant sexually transmitted diseases, but it's not a protozoan. HIV/AIDS is actually a virus.
Malaria?
Yeah, exactly. Malaria. Malaria throughout human history is the most devastating infection. It's killed more people than most other infectious agents. As a matter of fact, where I went to med school in Dominica, the campus that I went to was in Portsmouth. And then when I did my first clinicals, I went to Roseau. Roseau was the capital of the island. Portsmouth used to be the old capital of the island back in the 1700s. But during English colonial era, there was a malaria outbreak in Portsmouth, and they had literally had to move the entire capital of the island all the way down to the southern part of the island. So malaria is devastating. It's had that type of effect on lots of different regions in the world throughout history. So malaria is a protozoan infection. So Plasmodium falciparum is the most important one. There's also Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, but Plasmodium in general, that's going to be the most significant of all protozoan infections. Not so much here anymore in the US, but you do still see it a lot in places like sub-Saharan Africa and other parts of the world. Helminths. If you hear the word helminth, just think about worms, right? One way I like to think of helminths and the drugs that you treat helminths with is albendazole. Albendazole is going to be one of the most common anti-helminths. Bend, when you see the word bend in a drug, you're thinking bendy. I always thought bendy worms, right? And so we're going to talk about helminths a little bit later in this semester. Unfortunately, we don't really focus on it too much, but helminths are really, really interesting, and they do indeed fall under the category of microbiology, despite the fact that you can still see them with the naked eye. So here's a stereotypical eukaryotic cell. Make sure you know the different components. Like, for instance, I know it's kind of a funny meme, but the powerhouse of the cell, the mitochondria, you kind of have to know that. Endoplasmic reticulum, there's going to be the smooth and the rough endoplasmic reticulum, the Golgi apparatus over here, peroxisomes that break down different things, lysosomes. The only thing that you're going to see that's shared between eukaryotes and prokaryotes are going to be these, the ribosomes. Flagella. You're also going to see flagella too. But in terms of like things that are found within the cell, ribosomes are going to be basically the only thing. Ribosomes are going to be important for things like transcription and translation, right? So synthesizing proteins, you are going to see those on both. But know the basic, components of a eukaryotic cell. A lot of people get these wrong, unfortunately. But mitochondria, let's just talk about mitochondria very briefly, just because we're talking about the difference between prokaryotes and eukaryotes. There's a theory, it's called the endosymbiont theory of mitochondria. Endosymbiont. Now, the reason why it's called that is because endo, within, symbiont, symbiosis. You guys familiar with symbiosis? There's like mutualism, commensalism, and parasitism. Those are the different types of symbiosis. That's basically the way that different organisms interact. A lot of the bacteria we're going to talk about are going to be either mutualistic or they're going to be parasitic, right? So you think about like a parasitic organism, right? Where one benefits at the expense of the other. Now, mutualism is where both parties benefit equally. And it is thought that mitochondria used to be a prokaryotic organism. The reason being is because mitochondria have their own separate set of DNA. Mitochondria actually has circular DNA, which is what you find in bacteria. You find circular DNA in bacteria, and you find circular DNA in mitochondria. So the thought was that at some point in the past, a eukaryotic cell gobbled up a mitochondrion, and it said, oh, cool, like, you make energy, I'll provide you with, nutrients and shelter, and then we can have a, symbiotic relationship. And so that's kind of like an interesting theory. And I think it holds a lot of weight because it's very, I think it's a pretty legit theory in general. So in terms of major key components, cytoplasm, right? So that's going to be what you find on the outside of the cell. You're going to find the nucleus and the nucleolus. Nucleolus is where you find the nucleolus within the nucleus, and then you find ribosomes. But make sure that you keep in mind that both prokaryotes and eukaryotes have ribosomes. The main difference, though, is that eukaryotes have a different size ribosome. So eukaryotes have 80S ribosomes, whereas prokaryotes are going to have 70S ribosomes. Now, we're going to talk about why that's important, especially when you get into drugs. Once we get into drugs, there's certain drugs that target specifically the prokaryotic ribosome. So aminoglycosides are a good example. Macrolides are another example. Tetracyclines are another example. All those drugs are going to be specifically targeting the 70S ribosome, meaning that if you treat a patient with those drugs, it's not going to have an effect on the host as much as it's going to have an effect on the actual bacterial infection. There are going to be side effects, right? Every drug has a side effect, but it's going to be specifically targeting the 70S ribosome, so not as significant of an effect to the eukaryotic host. Make sure you know your mitochondria. We just talked about that. That's going to be the quote unquote powerhouse of the cell, as the silly meme goes online. The Golgi apparatus, where you see recombination of complex carbohydrates, lysosomes, which help to break things down, right? Lysis is going to be one of the major components of lysosomes. So cool. Let's talk about some of the major categories of eukaryotes. Do you guys have any questions so far? Okay, cool. So fungi is going to be one of the major categories of eukaryotic organisms. Fungi, arguably, I think, are one of the most interesting of all the eukaryotic organisms. Insofar as their biodiversity, right? There's a very diverse amount of fungi. Also, in the role that fungi play in the environment, fungi are going to be, by and large, saprophytic organisms. So here's that term, saprophytic. Saprophytic means that an organism that breaks down dead organic material. So it allows for the recycling of nutrients back into the environment. Fungi, if there weren't fungi, a tree would fall down in the forest and it would just stay there. The wood wouldn't do anything. It would just be inert. It wouldn't decompose. Fungi come in and actually help to break down those dead components, right? And so dead organic matter, and allows for that those nutrients to be recycled into the environment. So very important for that. Also, lots of really interesting fungal species that do all sorts of different things, some of which are good for industry, some of which are going to be potentially pathogenic. So here's an example of organisms that are good for industry. So like yeast. I know this is Malassezia furfur, but also a good single cellular yeast would be Saccharomyces cerevisiae. If you speak Spanish, Cerveza, Cerevisiae. Saccharomyces cerevisiae is what you make beer out of, right? Beer, wine, distilled spirits, also brewer's yeast, bread. So you can use single-celled yeast for making bread. So in other words, you have fungal organisms that are really important for industrial activity, but you also have fungi that are going to be potentially pathogenic. A good example of a pathogenic fungus is Aspergillus. Aspergillus, which is found in peanuts, for instance, like rotting peanuts. Have you guys ever heard of aflatoxin? If you guys haven't heard of that, that's fine. We're going to talk about fungus a little bit later, but Aspergillus produces aflatoxin, which can be potentially carcinogenic. So some fungus are good, some fungi are bad, but in terms of their biodiversity, a very interesting class of organism. We're not really going to talk about algae so much in this class. All you really need to know about algae is that they are going to fall under the eukaryotic, branch, and they are photosynthetic. So they're going to have chloroplast and chlorophyll. So they're in other words, they're autotrophs. Fungi are actually not autotrophs. They're heterotrophs. They require energy from outside, right? But autotrophs are able to create their own, energy through photosynthesis, and algae are going to be able to do that. So algae are kind of cool because they can exist in single cellular organisms, or multicellular organisms like what you find in kelp or seaweed. That's all algae. So very interesting. You can also see bioluminescence in algae. Like if you go out swimming, where I went to med school, for instance, in the West Indies, I would go swimming out at night in the ocean, and you would see the water just glisten every time you would, take a breaststroke. It's pretty amazing stuff. But in terms of this class, we're not really going to focus on algae. Protozoan, however, we are going to talk about protozoans. And there's something that's very unfortunate about when we do Zoom lectures, is that videos that I pull up don't really play. Let me see if I can get this to work. If I can't get it to work, then you guys are going to have to do this. Look at this on your own. Dang it. Yeah, so I don't know if I'm not gonna be able to get this thing to work. So whenever you guys get a chance, look at some of the links that I put in here. This one is really interesting. It's just like a ciliated paramecium. Paramecium are really cool. They're like, they live in the water, and they have all these cilia all around their body. So those cilia, they kind of like flutter, so they allows them to mobilize in the water. So very cool. So that's a paramecium with flagella. You also see other protozoans moving around the environment using things like, that was paramecium with cilia. So these are cilia right here. But you can also see flagella, right? So those are going to be like little tail-like projections that allow these organisms to move in the water. Protozoan are going to be single-celled organisms. They are eukaryotes, but they're going to be very simple, right? Simple, single-cellular type organisms. But they also have things like a membrane-bound nucleus. They have membrane-bound organelles. Diplomonads like Giardia, this guy over here, that's Giardia. If you were to look at this under light microscopy, you would see two giant nuclei, and it looks like two little eyes looking at you. It's called a diplomonad, and the diplomonad term derives from the fact that it has two nuclei. So you can see multiple nuclei in different types of protozoan organisms, depending on what they are. Now, helminths. This really sucks because I would love to play these videos for you. So what we're going to do is when we meet next week, I'm going to play these videos for you because they're startling to say the very least. So helminths, they do fall under microbiology, notwithstanding the fact that you can indeed see them with the naked eye. Like this is a liver fluke right here, big fat nasty worm. You can actually see that with your naked eye. They're actually remarkably large. This over here in that image on the top right, that's Ascarius lumbricoides. That is a very, very large nematode. It's huge, so big that they can indeed cause intestinal obstruction. And if it causes intestinal obstruction, what you would see in this video right here, this video will show you what it looks like when they surgically remove the worms from someone's colon. So it's awful. Let's just put it that way. I'll show you guys this in class next week. So these are nematodes, Ascarius lumbricoides. Tinea solium, those are going to be segmented type worms. There's two types. There's Tinea solium, Tinea saginatum, two major tapeworms. One is a pig tapeworm, one is a beef tapeworm. We're going to talk about that later on. But yeah, that's what you get from eating undercooked beef or undercooked pork. And then liver flukes, you can get that from all sorts of different things. One example is watercress. If you're eating watercress, you might get eggs on the watercress and you might ingest a liver fluke. They like to hang out around the duodenum because the duodenum is where you have bile getting released from your liver into your GI tract. And so I will show you that video next week when we meet up. So those are helminths. So let's talk a little bit about prokaryotes. So prokaryotes, as you guys recall, there's going to be your bacteria and your archaea, both of which have 70S ribosomes, no membrane-bound organelles, no nucleus, right? So you're just going to see the DNA free-floating within the actual cell, within the cytoplasm. And then instead of a nucleus, we call it a nucleoid. Now, the interesting thing about bacteria and prokaryotes in general is that the nucleoid is if you were to stretch out all their DNA, it would form a giant circle. It's a giant circular type DNA. Whereas with eukaryotes, we have linear DNA, right? So you have two complementary two complementary strands and counter-parallel going in opposite direction from one another. They do the same thing with prokaryotes, except they eventually wind up meeting at the end, right? So you have a giant circular DNA. Now with prokaryotes, and this is just a side note, but it's kind of important. So you have the nucleoid. So that's going to be the major component of the DNA in the bacteria. You also have these little tiny bits of small circular DNA, and those are called plasmids. And those plasmids are going to be generally non-essential bits of information. The most important information is going to be found in the nucleoid, right? But those plasmids can confer important things like resistance genes, resistance to antibiotics. That's going to be one of the most important plasmid type, genetic information. Those resistance genes can be transferred from individual to individual. And we call this conjugation. So that plasmid from individual number 1 here can be transferred to individual number 2 through conjugation. It's effectively the way that bacteria have sex. So they shoot out a pilus. That pilus is able to reach out to a donor, the recipient, and then the recipient is able to receive those plasmid DNA that can confer things like antibiotic resistance. So those are going to be very important. But just kind of keep in mind that there's 2 major components of the DNA in a prokaryote: nucleoid as well as the plasmids. And then you're going to find ribosomes, right? Ribosomes are very important for things like transcription, translation, etc. And then you're going to find certain things like capsules and slime layers. And we refer to those as quote unquote virulence factors. We're going to talk a lot about capsules and slime layers throughout the semester, but just kind of keep in mind that capsules are going to be things that you find on the outside of a bacteria that can confer not only drug resistance, not really resistance per se, but it makes them more I guess you can call it drug resistance. It makes it so that they're more resilient against drugs. It can also help them evade things like phagocytosis so that they're not as easily gobbled up by cells in the body. So we're going to talk about that later on, but those are going to be some virulence factors. Cool. So any questions so far? Okay, awesome. So let's keep moving on here. We talked about cilia in like paramecium, for instance, in those protozoan infections. Cilia you can also find in prokaryotes. Flagella are going to be also found in prokaryotes. Those are going to be those really large projections that allow for greater motility. You're also going to occasionally find spores. Now, spores, when you talk about spores in prokaryotes, don't confuse those with spores that you find in fungi, because in fungi, spores are going to be reproductive. Spores are what get released into the environment that allow for colonization of different substrates, right? Spores in bacteria are going to be a lot different. So if you have a bacteria cell and then you have a spore, the spore is the way in which a spore-forming bacteria can protect its genome. It can protect its DNA from harsh environmental conditions. We talked about Clostridium difficile already, right? So C. diff, that causes C. diff diarrhea. Clostridium organisms are going to be spore-forming organisms, not just C. diff, but Clostridium tetani, that causes tetanus, Clostridium botulinum, that causes botulism toxicity. Those are going to be spore-forming bacteria. And when they form a spore, it creates this kind of like proteinaceous, calcium-rich protective layer that protects the DNA from harsh environmental conditions. So in other words, a spore in a bacteria is not reproductive. That's the way in which the bacteria can actually undergo hibernation. So that they can lay dormant. You can find them in the soil. You guys familiar with tetanus, right? Clostridium tetani. You find it oftentimes in the soil, in the dirt, right? It's on a rusty nail, right? And so now if you step on that rusty nail, you introduce that spore into your tissue, and that's going to introduce that spore into a more hospitable environment. Then the cell is going to change from a spore into a vegetative cell. So we're going to talk about that a little bit later when we talk more about like Clostridium organisms. But just know that spores are going to be the ability for prokaryotic cells to undergo hibernation. Okay. And then these are some of the major components of bacteria. You're going to find things like fimbriae/cilia, cell walls. So the cell wall is going to be one of the most important differentiating factors, especially between gram-positive and gram-negative organisms. We're going to talk a lot about that later on. Pili or pilus, that's going to be that projection that allows for conjugation, where you transfer plasmid DNA from one bacteria to another. Flagella is what allows them for mobility, right? And cytoplasm, ribosomes, all that, then the nucleoid on the inside. So let's talk about the major components of bacteria. So we already talked about how prokaryotes, that means before the true nucleus, right? You don't have like a true nucleus. You have the nucleoid instead. So that's going to be one of the major components. And then you have multiple different types of shapes. We're going to talk about all these different shapes because it's going to be very important for the categorization of bacteria. You have rod shapes, you have spherical shapes, and you also have corkscrew type spiral shapes. We're going to talk about those today here. But rod, bacilli, spherical, cocci, and then spiral, you can refer to them as spirillum or spirochetes, depending on what the species are. One of the major components of bacteria are going to be peptidoglycans, and peptidoglycans are what make up the cell wall. Now, there's a difference between different classes of bacteria. So gram-positives are going to have a very thick peptidoglycan cell wall versus gram-negatives. They do have peptidoglycan, but it's going to be very thin. Okay. So we're going to talk about those details a little bit later. But peptidoglycan is going to be one of the major components of bacteria. One big thing that's important about peptidoglycans is that they can be targeted with antibiotics. You got any of you guys want to volunteer to take a guess at what antibiotics target peptidoglycans? What's like one of the most common?
Penicillin.
Penicillins. Yeah, excellent. That's great, man. So penicillins, that's going to fall under beta-lactams. Beta-lactam drugs specifically target the synthesis of peptidoglycan cell walls. And so if you use penicillin, amoxicillin, ampicillin, methicillin, all those drugs are going to inhibit the ability for the bacteria to form those peptidoglycans. Once they do that, once the bacteria starts dividing, it's going to explode because they rupture due to the osmotic pressure. The peptidoglycan helps to maintain their structural integrity of the bacteria. Speaking of
quick
dividing,
question, Dr. O.
Yes, go for it.
Just real quick, I know you're talking about the peptidoglycan. Does that change with different types of bacteria?
Oh yeah, absolutely. So gram-positives are going to have a very thick peptidoglycan. And we're going to talk about this a little bit later. But with gram-positives, they have this component called a teichoic acid scaffolding structure. So imagine peptidoglycan is horizontally structured, right? And it's going to be in multiple different layers, right? We're going to talk about this later on. Those layers are all going to be held vertically by teichoic acid. And those teichoic acid allow for the scaffolding of peptidoglycan. Gram-positive organisms, great example would be Staphylococcus aureus that you find on your skin, right? That causes abscesses, folliculitis. Staph aureus is a gram-positive, and it's going to have a very thick peptidoglycan cell wall. Same thing with Streptococcus organisms. So Streptococcus pyogenes, Streptococcus pneumoniae, all those organisms have very thick peptidoglycan. And then gram-negatives are going to have a very thin peptidoglycan. So a good example is E. coli. E. coli is a stereotypical gram-negative organism you find in your GI tract. It does have peptidoglycan for a cell wall, but it's very, very thin. So we're going to talk about.
So is that going to determine what medication can penetrate that wall?
Totally.
Depending
Absolutely.
on thickness or thinness. Okay.
Absolutely. Yeah.
Cool.
So gram negatives are very difficult to treat with traditional like beta lactam type drugs. So you have to use different types of drugs like trimethoprim sulfamethoxazole is an example. That's Bactrim. Have you ever heard of that in the hospital? So you're going to want to use different types of drugs for gram negatives for the most part. So gram positives are going to be easier to treat with their traditional beta lactam drugs. So like penicillin derived drugs are going to be really good for gram positives. So that's what you basically highlighted why I was highlighting the peptidoglycan structure, because it is very important. It's critical for antibiotic treatment. So that being said, binary fission. This is another important thing with bacteria. Bacteria do not undergo division like eukaryotic cells, right? They don't undergo mitosis or meiosis, right? Mitosis is what you find in your somatic cells in your body. Meiosis is what you find in your sex cells like gametes. Bacteria undergoes binary fission. That's where they synthesize all their DNA and then they eventually split apart. Whoops, what did I just do here? So, okay, so those are the major components of bacteria. Archaea, very, very similar to bacteria, except no peptidoglycans. They have different structures. They have different structures that allow for like structural stability that are going to be a lot more heat resistant and temperature resistant. That's what allows them to like exist in places like these black smokers, hydrothermal vents at the bottom of the ocean, etc. So that's what lends them to the ability of being extremophiles, right? But they do have a lot of shared components, right? They do not have a nucleus. They do undergo binary fission. Some do have flagella, and they have a 70S ribosome. Okay. So here's a chart that kind of breaks down some of the major differences between bacteria, right here, archaea versus eukaryotes, right? So nucleus, there's going to be no nucleus in bacteria or archaea. The archaea and bacteria are both going to undergo binary fission, right? Bacteria will have peptidoglycan, but archaea do not have peptidoglycan, both of which are going to have 70S ribosomes, right? No membrane-bound nucleus, no membrane-bound organelles, right? So those are going to be some of the major differences in bacteria versus eukaryotes. Bacteria and archaea versus eukaryotic organisms. Cool. So let's see, what time is it? It's 6:16, or I'm an hour behind you guys, so it's 7:17 over there. So let's continue on at around 7:30 your guys' time. I'm going to go ahead and do a quizzes. This is how I take attendance, by the way. Attendance in this class, of course, is going to be mandatory as per the requirements of Arizona College. And so the way I take attendance is I do quizzes. So you guys log in and then you answer questions. I'm not going to grade you on those. That's just I test you on the concepts, but that's how I input your attendance. So we'll do that towards the end of class today. Generally speaking, I'm going to start doing it spread out throughout the class, so it's not just all at the end, just to make things a little bit more interesting. But today we're just going to do it how I usually do it at the end of class. So anyways, so we talked about eukaryotes, we talked about prokaryotes. Let's talk about some organisms that don't really fall under the category of like living things, right? So viruses and prions. These, of course, can cause disease, right? But they are not going to be considered living organisms. Now, with viruses, the reason why they're not considered living organisms is because A, they're not able to self-propagate, right? They're not able to reproduce their own genetic information. In other words, they require a host. They have to hijack the host's replication mechanisms in order to actually reproduce their own, genome, whether it's RNA or DNA. Another thing that makes viruses non-living is that they're not able to metabolize, right? In order for something to be considered alive, it has to not only be able to reproduce itself, but it also has to be able to metabolize, right? So those are some of the major components of viruses. That's what makes them like non-living. Prions are going to be a little bit different. Prions do not have nuclei. They do not have nucleic acids. They're only proteins. Now, prions cause disease by causing a downstream cascade of misfolded proteins. You guys ever heard of like mad cow disease or Creutzfeldt-Jakob disease? Those are going to be some of the more common prion diseases. Other examples are bovine spongiform encephalopathy, Kuru. What's another good one? Familial insomnia. So familial insomnia is going to be another example. But we're going to get into prions later on. But just know that prions are non-living, but they're misfolded proteins. And those misfolded proteins cause basically a downstream cascade of misfolding. So they destroy tissue, mostly neurological tissue. So we'll talk about that later on when we get to viruses and prions. So here are a couple examples of viruses. Viruses can be all sorts of shapes. We're going to talk about the different shapes of viruses later on, but they can be naked viruses. They can be enveloped. So a naked virus is just the nucleocapsid, So it's got like this icosahedral type capsid with multiple different sides. This is a really crappy drawing. Pardon me, I'm using my mouse. But then if the nucleocapsid is surrounded by an envelope, that's what you got right over here. That's an envelope virus. So think about coronavirus. Coronavirus because it looks like a crown, like a corona. For those of you who like to drink Coronas. They're going to have an envelope that surrounds the nucleocapsid, and that envelope is made up of a phospholipid bilayer that is actually derived from the host cell. So those are going to be lipids plus the nucleocapsid, so it has nucleic acids as well as proteins as part of the nucleocapsid. But viruses can also be filamentous. So here over on the right side, those are going to be Ebola viruses. Those are filamentous viruses. It's basically just like a thin, long strand of a virus. So it's got a nucleocapsid that's basically a giant, thin strand. So we'll talk about that later. And then prions. Here's another image of what this looks like. So you have different shapes of your proteins, right? Once your proteins get formed, right, when you have your polypeptide getting formed by the ribosome, right, that's going to be your primary structure, right? It's just a one-dimensional primary structure. It's a strand of beads, right? It's a strand of beads of proteins. Once those strands of beads start interacting with each other, they start forming a secondary structure. So those could be pleated sheets, or they could also be alpha helixes. So that's going to be your secondary structure. So that's primary, secondary, and then tertiary structure is the actual functional structure of the protein. Tertiary structures can also form quaternary structures. So that's going to be this guy over here. That's going to be multiple different tertiary structures all combined together. Hemoglobin is a really great example of that. Hemoglobin has several different tertiary structures that combine together that allows for hemoglobin to do its thing, right? To be able to carry oxygen. What prions do is that they interfere with all these different structures, right? So they go in, they destroy those structures, they cause protein misfolding, thereby making those proteins no longer functional, right? And so they can cause things like fibrils. And some great examples, Creutzfeldt-Jakob disease, bovine spongiform encephalopathy. Those are all going to be examples of prion diseases. We're going to talk about those later on. Cool. So we talked about pathogenic bacteria and organisms, as well as organisms that are part of our normal microbiota. So when we talk about our normal microbiota, we can refer to it as normal microfauna. We can refer to it as normal, your normal microbiota. For instance, think about your gut microbiome. Those are all going to be terms that you can use to describe the things that are like, healthy, that are good, things that are useful for your day-to-day metabolism and activity. Places in your body that you should never find any organism are going to be blood and CSF, so your cerebral spinal fluid. It used to be thought that urine was also sterile. It's actually not really thought to be sterile anymore. We now know that urine is always going to have trace bacteria, not to the point that it results in a urinary tract infection, but it's always going to be a little bit of bacteria in the urine. You are never supposed to find bacteria or any organism in the blood or the CSF. If you get it in the blood, that's when you become septic, right? So septicemic, that's going to be where you have bacteria in your blood. You can also become septic if you have fungus in your blood. I had a patient, for instance, she had candida in her bloodstream. Candida is something that you find normally in your oral mucosa as well as vaginal and like anal mucosa. So she had candida in her bloodstream. Anyone want to take a guess why she had candida in her bloodstream? I'm going to talk about this later on when we get into infectious endocarditis and sepsis. But this is a patient that I had that was, she had Munchausen disorder. Have you guys ever heard about that, Munchausen? So there's both Munchausen and is Munchausen by proxy. Munchausen is where you feign an illness so that you get attention from people, right? So you make yourself sick so that you get attention from doctors, nurses, family members, friends, etc. Munchausen by proxy is even worse than that. It's like where you feign the illness of someone close to you. Usually it's like a parent and a child. Usually like they'll make their child sick so that their child is ill, so that the child gets attention from like healthcare providers and doctors and whatnot. It's like a really weird, strange way for people to get attention, right? So it falls under like psychiatric conditions. My patient had Munchausen disorders, and so she was making herself sick. And so what she was doing was she was spitting into her IV line. And because you find candida in your oral mucosa, she was introducing candida into her bloodstream. And so what happened is that the candida eventually found its way into her heart valves, caused a vegetation, so caused infectious endocarditis. She got a septic embolus and got a stroke and passed away. And so you can get septic, not just from bacteria, but you can also become septic from things like fungus as well. So long story short, not just bacteria, also fungi in your blood. Sad story. CSF, we're going to talk a lot about that when we get into bacterial meningitis as well as viral meningitis. And we're going to talk a little bit about that with encephalopathy as well. So we'll talk about CSF much later in the semester. It's week 7, so chapter 8. So we'll talk about that much later on. So here is a chart from your textbook that talks about different areas in your body where you do find different types of bacteria, where they can be pathogenic or they can be normal. So in your throat, for instance, you have a lot of bacteria in your mouth, most of which are going to be streptococcal organisms. You can also find streptobacilli as well. Like lactobacillus is a very common thing that you can find in your mouth. We'll talk about that later on. But Streptococcus viridans, strep mutans is going to be the bacteria that falls under strep viridans. Those are going to be the ones that cause dental caries, aka cavities. You find that very commonly in your mouth. Dentists back in the day used to commonly prescribe antibiotics before a routine dental procedure. They don't do that anymore now because of antibiotic resistance. But the reason why they did that before like a routine dental procedure is because of strep viridans. If you had really crappy teeth, poor dental hygiene, and you accidentally cut your mouth, you could potentially introduce bacteria into your bloodstream that could get to your heart and cause infectious endocarditis. So that's one of the biggest concerns for dentists back in the day. Now they only do that for patients that have a underlying heart condition, like a heart murmur, or if they have like prosthetic heart valves. That's when you get dentists actually prescribing antibiotics before dental procedure. But you find all sorts of strep in your mouth. You also find strep pyogenes. Strep pyogenes, it's a pathogen, obviously, because it causes strep throat and it causes a lot of other things. We're going to have a whole section on strep pyogenes later on. Strep pyogenes can cause necrotizing fasciitis. It can cause strep throat. It can cause impetigo. It can cause all sorts of different diseases, including like things like toxic shock syndrome. A lot of people do have strep pyogenes in their mouth naturally. What I do at GCU with one of my labs is we do a throat culture. We'll actually like do a culture on a blood agar. And so you can actually culture different types of streptococcus. If you see beta hemolytic strep, that's strep pyogenes. And I would say about 30% of my students have strep pyogenes naturally occurring in their mouth. Why do they not get strep throat? Because they have their healthy microbiota, right? That helps to keep those bad bacteria at bay. Haemophilus influenza is something else that you can find in your oral mucosa. That's what causes otitis media, causes what else? Epiglottitis. It can cause meningitis. It can cause a lot of different infections. But you find it in your oral mucosa, but it doesn't really cause disease for the most part because you have good bacteria. So let's kind of fly through some of these bacteria that you find in your stool. Enterobacteriaceae, you find Escherichia coli, Klebsiella. We're going to talk about Klebsiella later on when we get into pneumonia. One of the second leading causes of pneumonia is Klebsiella pneumoniae. It's usually caused by people who vomit and aspirate. So it happens a lot to alcoholics. So I usually call it, getting drunk in the club. Klebsiella pneumoniae is going to be the second leading cause of pneumonia, especially amongst alcoholics. Things that are pathogenic in your GI: Giardia, C. diff, Clostridium difficile, Salmonella. We're going to talk about that later on. We're going to get into the neurological disorders in chapter 8, week 7. So we're going to talk about Neisseria, meningitis, strep pneumo, and Haemophilus, and then all the different things you find in your sputum. By the way, where is it at? I want you guys to know that staph epidermidis, this guy right here, highlight this in your notes, because that is something you find in your sputum, aka when you cough up. Two things you find in your sputum: you can find Staph aureus, and you can find Staphylococcus epidermidis. Now, the thing that's confusing about staph epi is that epidermidis, where do you find it? You find it mostly on your skin, right? Your epidermis. But you can also find it in your sputum too. This is going to be on your exam. That's why I want you guys to highlight that. We're going to be culturing staph epi for our gram staining lab, so you guys will be able to actually visualize those underneath microscopy later on. Cool. So, any questions so far? We are now at 7:30 for you guys, 6:30 my time. Let me see where we're at here.
Can you go back to, I know it's like a little bit far away in the beginning of the slide, but the 70 and 80s?
Yeah.
Like, can you explain that way more? Because I just don't understand, like, does that, how many, like, I don't say that's how many it has, but
Oh,
what do you mean by just 70, 80?
Oh,
like,
it's
the level, like what's
I know what you're saying here. So it's that's how they categorize the size.
Okay.
Yeah, so it's the size of the actual ribosome. Where the heck is this image? So for instance, here's ribosomes, right? A ribosome looks like a ribosome, like, it's just like a tiny little bit of protein or mRNA, ribosomal RNA, sorry. And then there's going to be a large subunit and there's going to be a small subunit. And the entire both combined are going to be either 70S or 80S. For bacteria, it's going to be a 70S size, right? And then it's going to be made up of a 50S large subunit and a 30S small subunit. Now, don't ask me why the hell they named it that way, because obviously 50 plus 30 is 80, right? But for bacteria, it's going to be 70S. When you combine those two together, it makes a 70S subunit. I don't know why they named it that way. I should probably look into it, to be honest with you, because it always boggles my mind. But different drugs are going to target specifically either the 50S or the 30S. So for instance, like the 30S aminoglycosides are going to target the 30S. If I remember correctly, macrolides target the 50S subunit. We're going to get into the antibiotics later on. But that's what I mean when I said 70S versus 80S. In our cells, the eukaryotic cells, you're going to see an 80S subunit in our cells. Sorry,
Okay,
not an 80S subunit. It's
so
going to be
if
an 80S ribosome.
you measured the size, for instance, and you said it was 30, then you would prescribe the drug.
Yeah,
But
so
what if you can't see it? Like, what if there's just something, like, I mean, what if you go to the doctors and you have like,
all bacteria are going to have 70S.
no matter what?
No matter what. Yeah, exactly. So if you have E. coli, if you have, Klebsiella pneumoniae, if you have Staph aureus, they're all going to be 70S ribosomes. So that's the thing that's shared amongst all prokaryotes is the 70S ribosome, even amongst archaea. So bacteria and archaea both are going to have that size ribosome. And so that's why it's a good target for drugs, is because only bacteria are going to have that, right? Your mitochondria do have a 70S ribosome, though. I will say that. So certain drugs can affect your mitochondria. So that's one, downside to using those types of drugs. But for the most part, you're mostly going to be targeting bacteria using drugs that target that 70S ribosome. Does that make sense?
Yeah, it makes more sense for sure. I didn't understand when you were like 70S. I was like, what's that? Like how much?
Yeah, no, I get that.
How, like it just was like so blatant. I was like, what are you?
Yeah, if you look up ribosomes in general, there's multiple different components of a ribosome. I can't off the top of my head name off all the different components, but like by and large, a eukaryote 80s, prokaryote 70s.
I was just curious because I would obviously be like a test question, like, what's the size?
Oh yeah.
And then
Oh,
I'd be
totally.
like, well, it's like, what do you mean? Like 100? It'd probably say like 1,000, and I'd be
No,
like,
totally. You guys are going to be tested on that for sure.
Yeah,
You're going to be tested on the ribosomes. But the major component of the ribosome, you're not going to be tested on, like, I think there's like a 24 subcomponent of a ribosome. I don't remember, but 70s versus 80s, just know that and you'll be good. Cool. So let's talk about infections. We could do one of two things. We could talk about infections tomorrow when we get into before our lab. Oh yeah, by the way, this is how I structure my labs. Usually there's too much material to go over for one day of lecture. And so what I usually do is I use that lab day to piggyback off of the previous lecture. So in other words, if we stopped here today, we would continue on here tomorrow, and then you guys would go off and do your virtual lab. Does that make sense? I think some of your other classes do the same thing as well. But so with that being said, it's 7:36. Do you guys want to go ahead and do some questions?
Yeah,
If you have
let's
any
do it.
questions afterwards, yeah, sound good? Awesome. Let's do that then. So we're going to continue on with infections later on tomorrow. And let me stop screen share. Hold on 1 second. Yeah, we're almost done with that chapter anyways. It's we're about 2/3 of the way through. So we'll finish the rest of it tomorrow. All right, let's go into quizzes. So let me go ahead and share that screen. Where are we at here? Share screen. All right. Do you guys see my screen? Yeah. All right, great. So go ahead and log in. There are currently 18 of you. So somebody left the class. So they're not going to, unfortunately, they're probably not going to get their attendance. So go ahead and log in. Like I said, this is how I keep attendance for this class. You can either do it on your phone or you can do it on your laptop.
All right, I'm gonna be honest with you. I ain't that tech savvy, so you're gonna have to explain that.
Oh, you're good. Is anyone? Oh, okay. We got one person signing in. So if you go to joinmyquiz.com, you can either go on that with your phone or you can like open it up in a different browser for your computer. And then you just type in that code right there, the 145878. And I just saw that one more person joined our class. So go ahead and log into quizzes. Once we get all 18 logged in, then we'll go ahead and start the questions. If any of you guys have a really hard time logging in for whatever reason, shoot me a message in the chat box and I'll make sure I'll take note of your name. So I'll go ahead and put that into the attendance sheet. How's all your night? How's all your evenings going so far? Good? Anyone got any plans for the night or just laying low?
Dinner and bedtime.
You're at bedtime. Does anybody work nights in the hospital?
Not in a hospital,
but not in a
but
hospital. Yeah.
not in a hospital, but yeah, in a group home.
Oh, in a group home. Oh, okay. Fun times. Just
I might go over to Desert Vista every now and then and work a psych shift, but not too often these days.
nice. Nice. All right, so we have 16. A couple other people need to join. Who's having trouble? If you want, you can go ahead and put your name in the box, the chat box for Zoom. And then we can begin. Sony, you're having trouble logging in? Let me go ahead.
Sunny, actually.
You're having a hard time? Okay, I'll go ahead and screenshot so I got you in here. All right, cool. So with that being said, let's go ahead and start. We'll do as many questions as we covered. So I think I have like about 8 questions in here. So maybe we'll do about like 6 because I think that'll cover most everything we talked about today. So let's go ahead and begin. Has anybody in here used light microscopy in other classes? Have you guys used light microscopes? Who has not used a light microscope? Everyone's used a light microscope. Okay, good. So yeah, light microscopy is going to allow you to visualize things up to 1,000 times. So you have your ocular lens, which is what you look into with your eye, and then you have your objective lens. The objective lens goes up to 100x. Your ocular lens goes 10x, right? So you 10 times 100, you get 1,000, right? So that's going to be the actual size of the object that you're looking at. Okay, hold on 1 second. There we go. Awesome. Good. Most of you guys got that correct. Not all microorganisms are going to cause disease. In fact, the majority of microorganisms do not cause disease. Thank God, right? Otherwise, we'd be in a lot of trouble. There's a handful that do, and those are the ones that we worry about when it comes to medicine. So good job, guys. Oh yeah, by the way, when I structure these questions are going to be very, very reflective of what you guys are going to see on your exam. In other words, if you do really well on these questions, that means you're probably going to do pretty well on the exam. So just keep that in mind as we do these quizzes throughout the rest of the semester. So yeah, when it comes to referring to your good bacteria, all those terms are going to be fair game. You can use normal microbiota, normal flora, the microbiome. Think about the gut microbiome. Those are all going to be terms that you can use for the same thing effectively. So that is going to be on your test. Excellent. Most of you guys got this one correct. That's perfect. So membrane-bound organelles, that's going to be one of the key differences between prokaryotes and eukaryotes. And then the ribosome, 80S, eukaryote, 70S, prokaryote. All right, so there's quite a split on this question. For those of you who answered true, what was your thought process? If anyone wants to volunteer, you don't have to if you don't want to, but
Misclick.
Oh, go ahead.
Oh, it's just a misclick.
oh, it was a misclick.
Oh,
Okay. Anyone else have like any reasoning why they answered prokaryote versus eukaryote in terms of complexity?
I didn't answer that, but I could see why with the bacteria, the peptidoglycan, and I just thinking of complexity. Yeah, I guess it could go in either way.
Oh, I see what you're saying. Okay, that's a really good point, actually. When it comes to complexity, it's arguable that eukaryotes are going to be the most complex, right? Because they have multiple different membrane-bound organelles inside. They're all interacting in different ways, engaging all sorts of different types of functions. Whereas with bacteria and, archaea, they're going to be much simpler, notwithstanding the fact that there's a lot of variation, right? So don't confuse biodiversity and variability with complexity. Because eukaryotes, in terms of their physiological function, are going to have a lot more complex physiology and metabolism than bacteria. Even take, for instance, like respiration. In eukaryotes and prokaryotes, they can both undergo anaerobic respiration, but in eukaryotic cells, you're going to see a lot more aerobic respiration. You're going to see aerobic respiration as well as in bacteria too. But in terms of, like, for instance, let's look at yeast. Yeast are going to primarily engage themselves in anaerobic respiration. So yeast, they take sugars, right? And they break down those complex sugars into alcohol. That's going to be a very fundamental thing that yeast does. That's going to be an anaerobic process. The reason being is because there's not really much going on in terms of mitochondrial function. Whereas with the mitochondria, and especially eukaryotic organisms, that's where you see the electron transport chain, and that's where you see the development of like ATP, like a lot more ATP than you would develop under anaerobic conditions. And so in terms of just complexity of metabolism, you're going to see a lot more complex metabolism in eukaryotic species. But yeah, you're right. Prokaryotes do have a lot of diversity. So there's the ones that have the peptidoglycan wall. Some have mycolic acid. Some have, different components. So yes, there's a lot of variation. But just think eukaryotes are going to be a little bit more complex in general. Any other comments? All right, let's do one more here. It's kind of a silly question. I hope all of you guys get this one correct. Okay, good. Most of you guys got that correct. So eukaryotes are all those, right? Plants, fungi, protists, aka protozoa, animals. Those are all going to be eukaryotic organisms. Let's just do the rest. We'll get through it. All right. So that was kind of across the board here. So protozoans have membrane-bound organelles. So a lot of you guys answered sometimes. So let's talk about protozoans real quick. We briefly discussed those. A lot of those are going to be, well, let's define what a protozoan is. Proto means before, zoa means animal. So in other words, before it was a true animal, but they're animal-like. They're very similar to animals. Parameciums are going to be very similar to animals. They have like little cilia. I mean, if you watch them like, swimming through the water, they look like little dogs, they're kind of cute. Giardia, they have like flagella, so they like swim around the water. They have a lot of these like animal-like qualities, but they're not like true animals, right? So they're going to be considered before animals, but because of that, they're going to be considered eukaryotes because they have membrane-bound organelles. They have a true nucleus. They have all the components that make up, what an animal really is. They are all going to have different features and different structural, things that define them, but they're going to have membrane-bound organelles because they're all eukaryotes. So just make sure that when you hear the term protozoa, you're thinking something that's like before animal. It's it's animal-like in their nature, and they're super cool. It's a very vast array of species that fall under protozoans. Some that cause disease and some that don't. All right, let's do the last one here. All right, half and half, basically, almost, sort of. So viruses and prions are considered living organisms. They are not, right? Because first of all, prions, what's a prion? It's a misfolded protein, right? It's a misfolded protein that causes a cascade of misfolding downstream. Viruses, they don't cause misfolding, right? But it's like nucleic acids that are confined within a nucleocapsid. It's just basically a genome, but they're not able to reproduce their own genome by themselves. They're obligate intracellular parasites, basically. They require hijacking the host's DNA or RNA replication cycle and process to be able to reproduce their own genome. And then they can actually reproduce the genome, assemble their capsids and proteins and whatnot, and then exit the cell. So we're going to talk about that in viruses quite a bit. So don't worry if that concept wasn't super clear, but that's it, guys. That's it for lecture number 1. And again, I want to thank you guys for being patient with me and doing Zoom. I usually like to meet you guys day 1, but I'm at this conference, so I'll be back on Friday and I'll see you next week. Anybody have any questions? Yes, go for it.
Yeah, are we going to do these little tests every time we do a Zoom?
We're going to do these tests every single time we meet.
Okay, because they're extremely conducive. So, I mean, they help.
Good. I'm glad to hear that. I designed these. Like, I literally pulled up the exam and I kind of these aren't word for word, right? They're not exactly what you're going to see on the test, but the concepts are what you're going to see on the tests. So I took those concepts and I created questions so that they're going to be reflective of what you're going to see on your midterm as well as your final exam. But
I have a question.
Yeah, what's up?
How do we get access for all those questions that we just went through? Do we have access
yes,
to that?
I'm going to post a link to this later on. So I'm going to post a link to this probably tomorrow. I'll post a link for this version. Sorry, this set of questions, and then the next set of questions for tomorrow. And then you'll have access to those. So I'll give you these questions before your exams. Good question, though. Any other questions? Awesome. So tomorrow, we're going to meet. We're going to do the rest of chapter 1, and then we're going to do a quizzes again, and then just do your lab. Let me know if you have problem accessing any of the modules in Canvas. You shouldn't, but if you do, just let me know, and I'll try to resolve that. But other than that, I really look forward to meeting you guys in person. So it was nice meeting you virtually. So awesome. Have a great night. And if you need to reach me, shoot me an email or shoot me a direct message in Canvas.
Thank
Deal.
you.
Hey, good night.
My pleasure. Good night, guys. Have a great night.
Thank you.
Yep, my pleasure.